Life, in all its forms, is a journey. From the smallest insect to the largest whale, every living organism goes through a predictable cycle. We are born, we grow, we reproduce, and ultimately, we age and die. This is the natural order of things. But have you ever wondered why? Why do we age? What determines our lifespan? These questions have captivated scientists and philosophers for centuries. The answers, it turns out, lie deep within our cells, in the very blueprint of life itself. Our bodies are made up of trillions of cells, each containing a copy of our DNA. This genetic code dictates everything about us, from the color of our eyes to our susceptibility to certain diseases. And as we age, so do our cells. But cells are constantly dividing, creating new, identical copies. So why don't we stay young forever? Why does our skin wrinkle, our bones weaken, and our organs eventually fail? It's as if there's a hidden timer within us, counting down the days of our lives. This timer, as scientists have discovered, is very real. It's called the Hayflick Limit. In the 1960s, a young scientist named Leonard Hayflick made a groundbreaking discovery that challenged the prevailing dogma of his time. His work would go on to reshape our understanding of cellular biology and aging. Until then, it was widely believed that cells grown in a lab could divide indefinitely, given the right conditions. This belief was rooted in the idea that in a controlled environment, cells could escape the limitations they faced within a living organism. But Hayflick's experiments with human cells told a different story. His meticulous observations and rigorous experiments revealed a surprising truth. He observed that normal human cells, when grown in a culture, would divide a finite number of times before stopping. This was a stark contrast to the previously held belief of endless cellular division. This limit, now known as the Hayflick limit, averages around 50 to 70 divisions for human cells. Each division cycle brought the cells closer to a state where they could no longer replicate. After this point, the cells enter a state of senescence, where they remain metabolically active but lose their ability to divide and replicate. This discovery was revolutionary. It suggested that cellular aging was not just a result of external factors, but an intrinsic property of the cells themselves. It implied that our cells have an intrinsic limit on their lifespan, a biological clock ticking away from the moment we are born. This internal clock is a fundamental aspect of our biology, influencing how we age and how long we live. This had profound implications for our understanding of aging, suggesting that it's not just wear and tear, but a programmed process, deeply embedded in our cellular machinery. The shortening of telomeres, the protective caps on the ends of chromosomes, plays a crucial role in this process. The Hayflick limit varies between species, suggesting a link between this limit and an organism's lifespan. This variation indicates that different species have evolved distinct mechanisms to regulate cellular division and aging. For instance, cells from a mouse, with a shorter lifespan than humans, divide far fewer times in culture than human cells. This difference in division rates reflects the shorter lifespan of mice compared to humans. This correlation further strengthened the link between the Hayflick limit and the aging process. It highlighted the importance of cellular mechanisms in determining the lifespan of an organism, providing a deeper understanding of the biological clock that governs our lives. Imagine our DNA, the genetic blueprint of life, as a shoelace. At the end of each shoelace, we have a plastic tip called an aglet. This aglet prevents the shoelace from fraying and unraveling. Telomeres, in essence, are the aglets of our chromosomes. Chromosomes are thread-like structures found in the nucleus of every cell. They carry our genes, which are segments of DNA that code for specific proteins and determine our traits. Telomeres are repetitive DNA sequences found at the ends of these chromosomes. They act as protective caps, preventing the ends of chromosomes from sticking to each other or degrading. Each time a cell divides, its DNA must be replicated to ensure that each new cell receives a complete set of chromosomes. However, the DNA replication process is not perfect. With each division, a small portion of DNA at the very end of the chromosome is not copied, leading to a gradual shortening of the telomeres. Think of it like this. Every time the cell divides, it's like removing a tiny piece from the end of the shoelace. Initially, it might not seem like a big deal, but over time the shoelace gets shorter and shorter, and eventually it becomes unusable. Section 4. Telomeres and aging, a complex relationship. The relationship between telomeres and aging is complex and multifaceted. Telomeres, the protective caps at the ends of our chromosomes, play a crucial role in maintaining the integrity of our DNA. As we age, these telomeres naturally shorten, leading to cellular aging and eventually cell death. While telomere shortening is a natural part of aging, the rate at which they shorten can be influenced by a variety of factors, both genetic and environmental. Scientists have discovered that our genetic makeup can determine our initial telomere length, which can vary significantly from person to person. Our genes play a role in determining our initial telomere length and how quickly they shorten. This genetic predisposition can be a significant factor in our overall lifespan and health. For instance, individuals with longer telomeres at birth tend to have a longer lifespan and are less susceptible to age-related diseases. Some individuals are born with longer telomeres than others, and these individuals tend to have a longer lifespan. This genetic advantage can provide a buffer against the natural wear and tear that occurs over time, allowing these individuals to maintain better health as they age. This genetic predisposition highlights the inherent variation in the aging process among individuals. However, it's important to note that genetics is only one piece of the puzzle. Our environment and lifestyle choices can also significantly impact telomere length and consequently, our aging process. However, our environment and lifestyle choices can also significantly impact telomere length. Engaging in regular physical activity, maintaining a balanced diet, and practicing stress management techniques can all contribute to healthier, longer telomeres. Factors like stress, smoking, lack of exercise, and poor diet have all been linked to accelerated telomere shortening. These negative lifestyle choices can create a hostile environment for our cells, leading to increased oxidative stress and inflammation. These factors can create oxidative stress and inflammation within the body, damaging DNA and accelerating the aging process. Oxidative stress in particular can cause significant harm to our cells, leading to premature aging and a higher risk of chronic diseases. For instance, chronic stress has been shown to increase the production of cortisol, a hormone that can damage telomeres. Elevated cortisol levels can lead to a cascade of negative effects on our health, including impaired immune function and increased susceptibility to illness. Similarly, smoking exposes our cells to harmful toxins that can directly damage DNA and accelerate telomere shortening. The chemicals in cigarettes can cause extensive damage to our cells, leading to a faster aging process and a higher risk of developing age-related diseases. Conversely, a healthy lifestyle, characterized by a balanced diet, regular exercise, stress management techniques, and adequate sleep, can help protect telomeres and slow down the aging process. These positive lifestyle choices can create a supportive environment for our cells, promoting overall health and longevity. These lifestyle choices can reduce oxidative stress and inflammation, promoting cellular health and longevity. By making conscious decisions to lead a healthier life, we can positively influence our telomere length and, in turn, our aging process. Therefore, while our genes provide a blueprint for our lifespan, our environment and lifestyle choices play a significant role in determining how quickly we age by influencing the rate of telomere shortening. This interplay between genetics and lifestyle underscores the importance of taking proactive steps to maintain our health. This understanding empowers us to take control of our health and potentially extend our healthy years. By focusing on positive lifestyle changes, we can support our telomeres and promote a longer, healthier life. Section 5. Telomere shortening and disease. The gradual shortening of telomeres doesn't just contribute to the outward signs of aging, like wrinkles and grey hair. These visible changes are just the tip of the iceberg when it comes to the impact of telomere shortening on our bodies. It also plays a significant role in the development of various age-related diseases, which can drastically affect our quality of life as we grow older. As telomeres shorten and lose their protective function, our cells become more susceptible to damage. This cellular vulnerability can lead to a cascade of negative effects throughout the body. This can lead to a decline in tissue and organ function, increasing the risk of various age-related conditions. The impact is not limited to just one system, but can affect multiple organs and tissues. For instance, telomere shortening has been linked to cardiovascular disease. The heart and blood vessels are particularly vulnerable to the effects of aging at the cellular level. The cells lining our blood vessels, called endothelial cells, play a crucial role in maintaining vascular health. These cells are essential for the proper functioning of our circulatory system. However, as these cells age and their telomeres shorten, they become less effective at repairing damage, leading to inflammation and plaque buildup in the arteries. This increases the risk of heart attacks and strokes, which are major causes of morbidity and mortality worldwide. Similarly, telomere shortening has been implicated in neurodegenerative diseases like Alzheimer's and Parkinson's. These diseases are characterized by the progressive loss of brain cells, leading to severe cognitive and motor impairments. These conditions are characterized by the progressive loss of brain cells, and studies have shown that individuals with shorter telomeres may be more susceptible to these devastating diseases. The link between telomere length and brain health is an area of active research. Furthermore, telomere shortening can even play a role in cancer development. While it may seem counterintuitive, as short telomeres can lead to cell death, they can also trigger uncontrolled cell division in some cases. While it may seem counterintuitive, as short telomeres can lead to cell death, they can also trigger uncontrolled cell division in some cases. This paradoxical effect is due to the complex nature of cellular aging and the DNA damage response. This happens when the DNA damage response malfunctions, allowing cells with critically short telomeres to bypass normal growth controls and proliferate uncontrollably, leading to tumor formation. This uncontrolled growth is a hallmark of cancer. Therefore, telomere shortening is not just a harmless side effect of aging. It is a critical factor that contributes to a wide range of age-related diseases, highlighting the importance of understanding and potentially manipulating this process for therapeutic interventions. It's a critical factor that contributes to a wide range of age-related diseases, highlighting the importance of understanding and potentially manipulating this process for therapeutic interventions. Research into telomere biology could pave the way for new treatments that target the root causes of aging and age-related diseases, offering hope for healthier aging in the future.